counseling here in psychology. Hey guys, today we're going to be going more into detail of the way that your senses work. We're going to make sure that we understand your eyes, how vision works, and we're going to go into the auditory and how your ears work. Here's a picture of a rhino. It's really good. So, these are probably our two most used senses, so we're going to spend a fair amount of time on them. Vision is the one that we were most dependent on, and vision is dependent, and then auditory is probably the other one we notice the most. So these are our two most important senses, so I figure we probably need to know how they work, and there's some weird stuff that happens with this, but we're going to look a lot at the anatomy of the eye. So if you've had an anatomy class, you're going to be in better shape. And I'm not going to try to draw an eyeball because that's really hard. So I'll just put my eyeball up here and gross you all out. Okay. In your eyeball, you have a couple things. You've got photoreceptors in your eye, and they are the things that are sensitive to light. We'll go more in detail on that in a minute. And of course, we have the visible spectrum of light right here from red to violet. We talked about that last time. It's pretty small, and that's what we can see. So let's go look at the eye. I have a picture of an eye here, and we're just going to go over what each part of the eye does. Where did I put this picture of the eyeball? Oh, by the way, I'm going to link this Radiolab thing as well. This is so cool. It talks about the ways that your eyes work. If you were wondering what would it be like if you had more rods and cones and stuff in your eyes, this talks about this. Also, there's this cool thing on how nobody knows the color of the sky was blue. If you have some time to listen to a really interesting podcast, listen to this. I'm going to link it in this thing. Listen to it. I'm not going to make it, but it's a lot of you had questions about the rods and cones in your eyes and what would happen if we had 4 instead of 3. Listen to this. It's so cool. I'm glad I found it. I didn't know if I'd be able to find it. Anyway, here's your eye anatomy. Here's what your eyeball looks like. I'll just make the rhino small. All right, so this is your eye. And I'm going to make this medium size. I'll put this over here, and we've got our nose over here. So there's your eyeball, and here are the parts of your eye and how they work. All right, you've got the sclera, which is the white part of your eye. That's the white. That right there, sclera. It's the white part of your eye. It provides protection to your eye from UV rays, and it gives it structure. Humans are probably not the only animal where we always see it, but it's the only one I can think of. Maybe there's another one, but human eyeballs are weird in that you can actually see it. The cornea. So the cornea is this thing right here, and it goes over your eyeball, and it's the prism by which light enters. And so this is, so it's kind of the covering on the eye. If you ever have LASIK eye surgery, they cut the cornea off so they can get to the stuff underneath. And then they shoot lasers. When they do that, so I had LASIK, and went like this, and then they took a laser and they around my eyeball and they flipped the cornea off, and then I couldn't see anything for a little while. And then they put it back on and I could see things. It was neat. I could see that the lasers, when they hit my eyes, it was like blue dots all over the place. Now we have the iris, this thing right here. And that's the pigmented muscle. It's a muscle in your eyeball. And it's not just there because it looks cool, but it helps you to control your pupil. So I have blue eyes. You guys are probably sick of looking at my eyes right now. But I have blue eyes. That blue part is called the iris. And the iris isn't there just to look neat. The iris is a muscle, and it helps to make the pupil open and close and that kind of stuff. It helps you to regulate that. And that's what the pupil is. The pupil is an opening in the iris that lets light in. When your pupils are really dilated, lots of light comes in. And when they are not dilated, it goes down. That's why they get bigger when you go into a dark room, because we need to have more light in. And it's why if your pupils are dilated and you go outside and it's bright, it hurts because you got too much light coming in. Then we have the lens. The lens is right here. And this is the shape covering convex structure that focuses images on the retina. So this is the thing that focuses it and helps you to make the image not blurry. When I had LASIK, they took the cornea off so they could shoot these things and change it because it was misshapen. So you change that, and then you can, that helps to focus it. So that's the thing that helps you focus on stuff. And you know how you can, if you look at a thing in the distance and then you change, you kind of control your eye and then you can turn it into, you can focus on it. That's the muscles in there working. Then you have the retina, which contains 2 types of photoreceptors. We'll talk about those in a second because they do very, very different things. And if you want to know why this will help you on the extra credit assignment that I've posted, rods and cones do different things. And so we're going to talk about those now. So these are the parts of your eye. The optic nerve, we're going to talk about, before we talk about rods and cones, we're going to talk about the optic nerve. So the optic nerve right here is the part that comes from your eyeball and goes outside the back of your eyeball and to your head. Now, oddly, right here, and we're going to talk about blind spots, and there's an assignment on blind spots. Right here, you see where this is? Since there's a physical thing there, light can't go there. And so you're kind of almost seeing your optical nerve when you have that blind spot. So if you go to the assignment, which is a real assignment, about and you look at things and you'll notice that there's a blind spot, that's because of the optic nerve. We don't have light going in there, so each eyeball has a blind spot. You actually have a really small hole, basically, in your vision at all times. And if something happens to fall right in where that hole is, and there's nothing there to help it out, then it will appear to disappear. Now, you don't notice this because both of your eyes are putting things together. It's called the Gestalt perception theory. And what it means is that our brains are kind of filling in blanks. You don't actually see everything you're seeing, you're not actually seeing. Your brain is going, oh, I have a blind spot here. Here's what probably goes there. It's so cool. Your brain, Coco, that's my dog. Shh. So super cool because you should. If you didn't do that, you would notice right about here, blind spots. Things where you couldn't see anything, but you don't. Gestalt perception theory. So that's this thing right here when we're talking about this. Both rods and cones, symptoms of bipolar cells, blah, blah, blah. The blind spot is where the optic nerve connects to the eye and contains neither rods nor cones. So there's nothing there. There's nothing to detect things. But you can see what's there, but that's only because your brain is filling in the blanks. It's filling in the blanks with stuff that isn't actually being seen. That's so cool. Okay, sorry, I got excited. Okay, let's talk about rods and cones. So let's go back to here. Pull up ye old picture of the rhino. And in your brain, you have rods and cones. So, you got rods. We'll say they look like that. And you've got cones. Let's say they look like that. These are inside your eyeballs. Now, rods are, they see movement, and they do not see color. Not see color. And they are generally, here's your eyeball, they are generally on the periphery. That means they are on the outside. So your rods tend to be more around the outside. This might be helpful to you for the people trying to do the extra credit assignment. Then you have cones. And cones are not necessarily for movement. So you have a few cones, you have a few rods here, but you mostly have cones. So here's your eyeball, right in the middle. Right here you have all your cones. And they deal with perception, colors. They tell us colors. You have 3 types in your head. You've got 3 different types of cones in your head, in your eyeball if you're not colorblind. If you ever wanted to know what it looks like the world looks like to a dog, ask a person who is colorblind because they have the same thing. A person who's colorblind has 2 sets of rods in their eyes. Dog has 2 sets of rods in their eyes, same type, and humans have 3. Some birds have 4, and I think mantis shrimp have 11. Listen to the podcast; it's so super cool. So these things are here, and they're not designed for perception. They're designed to see colors. So between these things, you can see colors and you can see movement. There's a reason why we have these things out here, because it's really important to be able to see stuff on the side of you in case it's trying to eat you. Peripheral vision is really important when it comes to survival. And it's important to be able to see the stuff in front of you, color vision, to see what it is. Color helps us to determine things in the world, helps us to see if a thing is dangerous or not, also if it's pretty or not. So, yeah. All right. What else should we go over? So, coding information in the retina. So the receptive field is where it sends, so here's our eyeball, and right here we have rods and cones. And these rods and cones connect to things, and they send signals out to what's called the ganglion cell. So a ganglion cell is just, it's like a neuron, and it kind of looks like this. It's gangly and all weird, and it's connected to all these things. So they're connected to this thing, and then that sends out a signal to your brain. I'm trying to go through this faster than I normally would. I'm not going to go over too much on the two general types of receptor cells. I'm going to try and go through this a little bit faster. Visual pathways from the eye. So there's a thing called the optic chiasma. So the junction of the two optic nerves, or the sides from the nasal eye side close to the nose, sides directly close the nerve fibers from the peripheral sides of the two retinas do not cross to the other side of the brain. The result is that the left half of the world is represented in the right hemisphere and the left half. So when it comes out of your brain, it's just going to, so like the right part of the right side of your brain controls the left side of your body. The right side of your brain controls the left side of your body. And so the things that you see, because it goes right back to the same place. So the things that you see aren't flipped. I guess is a way to put that. Anyway, they go back and you're controlling things kind of backwards. I'm not going to go for the visual cortex. Let's just go through color theory, because I find this. Why do we see colors? All right, so like I said, there are three cone cells. There's blue, there's green, and there's red. Sometimes these will be referred to as other things. So one responds really well to blue, one responds really well to green, one responds really well to red. And we perceive colors through a mixture of those things. It's like any type of, like a pigment thing. You mix things together and you get a color. And that is the Young-Holtzman trichromatic theory. And that's basically what's going on. If you are colorblind, you're missing one of those. Monochrome colorblindness, a person cannot see any color at all, which is pretty rare. And dichromatic colorblindness, the person receives only two of the three visual pigments. So if you're completely colorblind, then you're missing, it's something completely different. But if normal colorblindness is, you're missing one of these guys. Probably green, I think, because then you can't see the difference, you can't tell the difference between green and red. I think that is correct. Once again, the podcast is a whole lot better at that than I am. Problems that you can have. So you have your eyeball, you got cataracts. So here's your eyeball, and what happens is you get like it's almost like a scab. There's this stuff that built up on your eye in front of here, and just built up, or like if you've ever got calluses, it's almost like a callus, and it will blur your vision, and people can go blind from that. The surgery, it used to be a big giant pain in the butt. Like, you got to cut your eye open and cut parts of your eye off. They can do it with lasers now, do it really quick, and it's a pretty quick thing. Retinoplasty. Or retinopathy. So you have blood vessels in your eyes. your eyes are organs. So here's your eye. There's blood vessels. You can see them. Even if you're really tired or something, your eyes are bloodshot. Sometimes you can get damage to one of those things, and it might cause blurred vision, blind spots, or floaters. So sometimes if you see a floater or something, or if you do something that would pop one of those things, then the eye will pick up on that, and generally those things will come back. Like one time I was out running, and I think I had a small blood vessel pop in my eye, and I had a little patch in my eyeball wherever I looked, that part was blurry, and that was for about a week once. That was possibly retinoplasty. There's glaucoma. Now here's your eyeball, and like, imagine there's a needle, and it's pushing more stuff in there. You get a lot of liquid and fluid that builds up in the eye, and that pushes on and damages the optic nerve. So if you want to know what that feels like, take like right here, you have a nerve, and you push down on that. Now imagine that's in your eyeball. Glaucoma is really painful, and it's this pressure, this building pressure, inflammation from the fluid in your eye pushing on the optic nerve, and it causes people to go blind. It also causes people to sometimes see like rainbow type things, and it gives you blurred vision, and it's really quite painful. What you do is you try. There are some. I don't know exactly how to fix it. I've never had glaucoma, but I know a few people who have had glaucoma, and there are treatments for it. Macular degeneration. Macular degeneration. This causes the inability to see things clearly, distorted vision, dark spots in the center of the vision. So basically what this is so I talked about how the eye has muscles in it. So the muscles in the eyes start to degenerate, and they get weak, and so you can't focus your eyes. You can't contract or relax that muscle in a way to make it focus, and so things become very fuzzy, and it makes it very, very hard to see, and you get dark spots in the center of your vision. So you don't want macular degeneration. Some of you have this one, myopia. This is farsightedness, and this is where when you see things close, they get really blurry. I don't have farsightedness. As you get older, this one tends to get a little bit worse, and it's just because you cannot focus on things here. And one reason for farsightedness is a misshape of the lens. The lens has become kind of misshapen, or the material itself is less elastic. As you get older, people tend to get this more because it's harder and harder to focus on things. And then you have what I used to have, which is myopia, known as nearsightedness. Nearsighted. I spelled that wrong. Sightedness. And this is when you have a hard time focusing on things far away, and that generally occurs because the lens in your eyes is misshapen. You might also have an eyeball that is itself misshapen, and that can also cause things like that. So if you wear glasses, this one's more common in young people. But if things far away are hard to see, myopia. Okay, now let's talk about auditory stuff. So, audition, sounds. All right, so sounds are pressure waves. And that vibrated, and that vibration sent out a pressure wave into the air, and it made all the air molecules vibrate, and then it just passes along. It's like if you hit water and it creates a wave and just passes through. That is an energy wave going through, pressure wave going through water, and it's the same exact thing that happens in the air when you make sounds. So sounds are mechanical energy made by that's caused by vibrating objects. Or vibrating, so I hit a thing, it causes it to vibrate, causes the air to vibrate, bloop, sound comes to me. I don't have a thing for locating sound sources here, but there's a reason why you have ears on both sides of your head. And this is so you can detect where it is, because you can see which side of the head it's coming from. And so you listen to a thing, and I can tell that somebody in my house just makes noise, and they must be this way from me because I heard it over here. Now, if you are deaf, if you had your ear was like in your face, like right smack dab in the middle, it would be, and you didn't have this ability to have it on the sides, you wouldn't be able to tell where a sound source is coming from. Because what you're doing is, by listening to it and by where it is on your head, you're kind of triangulating where it is. It's here. Oh, okay. It's more on this side and less on this side, so it's over here. And then you listen to it, and eventually you can find that sound. Let's look up the ear. Okay, so we have a picture of the ear right now. Over here, this is the pinna. The pinna. This is that outside of your ear. It's that flap. And what it does is it helps to bounce sound waves into the auditory canal. So the auditory canal is this thing right here. It's like it's a hole in your ear, and it leads to what's called the tympanic membrane. That's the eardrum. So think of a drum. I don't have an actual drum in my house right now. So think of a drum, and the drum is skin or some sort of covering really tightly over a thing. And when you hit it, it vibrates. And that's what's going on. The sounds come in, they hit the tympanic membrane, and it helps to vibrate. So it separates the outer ear from the middle ear and vibrates with reception of sound. So it goes in and it vibrates. Then next to it are these bones. You have 3 bones on the inside of your ear. They're called the hammer, anvil, and stirrup, or the malleus, incus, and stapes. I'm going to go with hammer, anvil, and stirrup. And basically what happens is the tympanic membrane vibrates, which moves these things that transmit sound vibrations to the cochlea. So the cochlea is the inner part of the ear. It's this thing that looks like that looks kind of like a nautilus. It's like a shell, and here are tentacles coming out. And this contains cochlear fluid and cilia, which are tiny hairs in your ear. We'll talk about those in just a second. So this comes in and this vibrates this. When this is vibrating, this, which contains the cilia, the hair cells, the vibration of the fluid in here, so it changes, there's pressure changes in the fluid. So it comes in, vibrates, these things move, it changes pressures in here, which makes those hairs move. And when those hairs move, they're connected to your nerve endings right here. And that sends a signal down through your auditory nerves into your brain, which you can then figure out. Now, if you listen to a lot of loud, heavy things, those nerves, those hairs, can get permanently locked in a position, or they can break. And you can have that ringing in your ears thing. So if you go to a loud concert and you got that ringing in your ears, that's because the nerves have been pushed. Those nerve hairs have been pushed. And if it never goes away, they're kind of permanently bent in that direction, and they're constantly sending a signal of something to your head. And so you're hearing things that aren't actually there. It's generally a high-pitched wee. And if you've listened to a lot of metal music in your life, you probably have it a little bit. It's called tinnitus. So, what are the characteristics of sound? We have, let's pull this guy back up. Frequency. Frequency is pitch. So this is from here to here, how many things do we have apart? And that's going to be, how far do we have apart between these things? We talked about the Doppler effect before. So if you put more of them in one space, it goes up higher. This is pitch. And they're farther away, they're going to be lower. So a car comes towards you, it's sending out auditory waves, and it goes, and the room it's spreading out because the frequency changed. The amplitude is how high this went. So a really high one is going to be louder than a low one. This is so if I take one, and these two have the same pitch, but this one is louder than that one. Even though their pitch is basically the same because the frequency is the same. 1, 2, 3, 4. I got 4 in the same space. And complexity is how many various things are in there, and that's that wave function thing that we talked about. So there's some theories on how this happens. Place theory says that differences in pitch results from stimulation of different areas of the basal membrane. And there are other people who say frequency theory differences in pitch are due to rate of neural impulses traveling up the auditory nerve. So basically, the basic idea is, so how do we get pitch? So here is the tympanic nerve, and we have boom, 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 boom. Now, the place theory says that we get the pitch by where it hits on this. So different areas are stimulated in different ways. The frequency thing says that we have the same thing, and frequency, so this is where, so this hits in a different place and stimulates a different nerve, and frequency states that we get this different pitch because it's getting hit more often. I don't know which one is right. Personally, I think if I were to take a guess, I'd go that one because that's generally how nerve cells work. To change intensity or of a thing, it's mostly the how often you hit it. The difference between it's comfortable and ow that hurts is how intense and how often you're sending a signal. And if you're sending a signal with each time that this hits it, with each of these waves, then that would alter it to you, and that should probably change pitch. Although I do not know which one is right. That's why there are two theories, and my guess on that one. All right, so what can go wrong with you? You can get deaf. Being deaf sounds horrible. There's conductive deafness. And this is when when sound waves are unable to be transferred from outer to inner ear and cause it can be so. Here's your ear, the outer ear, and then to the inside ear, and there's something in here that makes it so sound waves cannot pass it. So they can't go from the outer ear to the inner ear. So why would this be? Well, you got gunk in your ear. It can happen with tumors, infections. If you've ever gotten an infection in your ear, it might be really hard to hear. Swelling, or degeneration of your middle ear bones. So those ear bones that we talked about, sometimes those can break down. And if they're not there, you're not going to be transferring the sounds to that thing that looks like to the cochlea, the thing that looks like the nautilus shell. Then there's sensory neural deafness. Now this is damage to the inner ear. So all this is okay, this is fine, the canal is fine, everything's okay here. We get to the inner ear and something is wrong. So you got the cochlea, you got those little hairs that are in there, and this could be oftentimes due to damage to this thing. Maybe this thing's hurt. A lot of times it will be due to these things being broken or not existing. And on these ones, the message is getting to these things, but they're unable to send it to people. Ways to get around this? Well, in this one, fix the thing that's blocking it if you can. And on this one, you can create things that will, like hearing aids work with this one, to increase the amplitude if it doesn't get to it, or to even send electrical signals themselves. I had a friend who had a hearing aid that he was completely deaf, and it was because of this, and they set it up so it sent signals to the auditory nerve, and he could hear, and it was really cool. Perception and attention change to make other incoming information more important to the lack of auditory import. For example, deaf people focus more on mouth movements, etc. So, I think I've talked about this before. Here's a brain. Now, our auditory senses are generally right here. And if they're not being used to hear things, your brain rewires itself. And this won't be an area that's not doing things. It's being used for other things. And so people who are deaf are oftentimes able to notice things visually maybe a little bit more, or they're more perceptive of motion or other types of things. And that's one of the reasons why you learn to do it, but you also have part of your brain that's now kind of retrained to do these things. It's true for pretty much any sense that you lose. You don't want to lose a sense, but generally when you lose a sense, your other senses do tend to become a little bit more acute. because it needs to, and that part of your brain is there. Unless, of course, it's because you hurt your brain, which is another reason you could go deaf, and that's if you damage the parts of your brain. And you could go deaf from brain damage, where everything in your ear is fine, but there's something wrong in your brain. Or, like, if the auditory nerves got cut, that could also cause you to go deaf. Anyway, I'm going to stop right now. I'm hoping that I kept this to a little bit shorter. Watch the podcast. It's really super... Or watch the podcast. Listen to the podcast. It's really, really interesting. You don't have to, but it's really interesting. I was spellbound. Do the other things to kind of keep track of it, and make sure that you send me your 3 questions, your things that you noticed or thought were interesting, and your summary at the end of this. I've been having a fun time reading yours. Thanks. See you guys later. Bye.